Okay, I think now we're ready. Okay, hi. So hello everyone. Again, had some issues starting the live stream here. Hello and welcome to another Saturday microscopy live stream. Today I'd like to put a few plant tissues under the microscope, and I'd like to show you some techniques on how you're able to cut them and how to section them so that we're able to prepare them for microscopy. I see that many people have already checked in, and you've already probably also noticed and realized that the setup, everything here again, is again a little bit different than from the previous videos. Last time I had my face in a nice little circle, but I received some comments that apparently you preferred if I have the regular traditional setup again here. I'm seeing that many people are already starting to comment, and I would like to ask you again, like all the time, can you please give me a short piece of information of whether you're able to hear me properly? Because I would like to make sure that the sound works. So if you are able to hear me properly, please tell me that the sound is good. Sound is good. Okay, good. So what I will be doing today, again, is I usually start off with a short 2-minute greeting session. I go down the chat here. Hello from Vietnam. From Argentina. Hello from India. That's nice. From California. Some people are learning Spanish. Hello from Tennessee in the United States, from Hungary. Okay, there is a little bit of my Spanish is practically non-existent. From England, a hello, and Mecklenburg in Germany. So yes, and I think that seems to be. And here's already the first question. Where can I get hold of some xylene, which you need for staining plant tissue? Honestly, xylene, you can buy it, of course, in a chemical shop, but xylene should be handled with very much care because it is not good for your health. So actually what you need to do is you need some kind of a fume hood, which actually sucks away all of the fumes of the xylene, because it's not good. Xylene can also be bought in some shops that sell paints, because it's also part of paint thinner. But I would be rather careful handling with this the xylene. Okay. So perfect. Sound is good. Sound is fine. Hello from the UK, from the Netherlands. Okay. So what I'm going to do now is the following. I've prepared a couple of plant leaves here. I have also wanted to show you a little bit some commercial cross section of plant tissues. So this means a little bit of biology today as well, where I would like to explain to you some of the things that you're able to see here. I do have my microtome. Some of you might already know it. But I want to make a couple of sections today also without a microtome, and also using a razor blade. As a matter of fact, as we 3D printed this plastic holder here, but you got to be really careful because those razor blades are extremely sharp. And today I actually bought some razor blades as well. I also bought one of those razors here, not because I want to shave myself primarily, but because I was experimenting on whether you can also use this razor to also cut sections of a plant. It does work, kind of, but I think it's still a little bit too thick. Okay, so I'll be also experimenting with this a little bit as a kind of a, how do you say, alternative to a microtome. As a matter of fact, I would like to start off explaining this here first, in case you don't know that. It looks a little bit old-fashioned, but it's a modern device, because some men like to prefer those razors over the other ones. And there is a little thing that you can turn here, a knob that you can turn here, and look how it opens up. Okay. And there is the razor blade in here, which is obviously extremely sharp. You basically put it in here, and then you close it again. And then I'm going to try to make some cuts with the razor to see if they actually work. Alternatively, I would like to, of course, try to use this one over here. We've got the same type of razor blade in here, but the plastic handle here is I've made myself using my 3D printer, because otherwise it's too difficult to hold and you might actually cut yourself. Yeah? So that's a little bit some of the things I'd like to try today. And as a little warm up, I just want to show you something. Something that is also quite nice. As a matter of fact, some months ago, one of my viewers actually watched the video and sent me some leaves. Okay, these leaves are actually from Thailand, because he wants to show me something here, and I'm going to take off out one of these leaves. It's completely dry. I don't know even from which plant it is, but there was something quite interesting on here that I would like to show you. Maybe, of course, you already know what I'm about to show you, because maybe some of you have already watched some of my previous videos. Yeah, but I'm going to now directly make a permanent mount of that, because actually I think it's quite nice looking. Okay, so I'm going to simply scratch off a little bit of this fuzzy material that you're able to find here. There is this very thin fuzzy layer, and the reason why I want to show this to you is because quite often you see in Reddit and online, in forums, people posting pictures wondering what this is. Okay, because sometimes you can actually see it also in water samples and in dust, especially, and so on. And I just want to show this to you. While I put it back here, I'm going to interrupt myself again, because some people are already posting again. So I like to do this every now and then, kind of to keep the flow going. Is where people post questions and I want to talk about it directly. Okay, some general comments. So I'm reading all of the comments, but I'm stopping at those where there is actually a question here. I went sampling last week and I found my first Dileptus and 5 planaria. Okay, planaria are flatworms. Okay. Hello from India. Yeah, pretty long question and also a little off topic. I wanted to ask you, should I buy the following microscope and accessories? Also, please a review video. Okay, so basically the question is which microscope to buy. I can talk about this separately as well. Honestly, it's almost impossible to say because it depends so much on personal preference. Yeah. Okay, so does the video look blurry to anyone else? Well, I hope it does not look blurry, because actually I do see it very clearly. So if it does look blurry, then it could be that you have a slower internet connection, and then YouTube lowers the image quality so that you're still able to get a smooth video. Okay, and then it can look blurry, but YouTube will try to kind of lower the image quality so that you get a smooth video if your internet connection is not fast enough. Okay, so that's a little bit thing, yeah. Okay, hello from Massachusetts, hello from India. All okay. So there's again a question about the video. So what I'm gonna do is, this is again my favorite PVA glue here. Okay, and I'm going to simply make a little permanent mount. Again, you got to be really careful with those permanent mounts, because if they're not dry yet, and if you then basically contact the microscope objective with the mounting medium, it's of course not so good for your objective. I'm going to do the following. I'm simply going to put it into the mounting medium, mix it a little bit to make sure that it's a little bit spread apart. I have to use a tissue paper. Again, like every week, I'm totally perfectly prepared and I cannot find a tissue paper. Yes, I did find one. Okay, and then of course a cover glass goes on top, and then we're going to have a look at this, and then maybe some of you might recognize the structures there, because maybe some of you, if you're into microscopy, have already seen those in dust samples, maybe even water samples, mostly in dust, I would say. So let's compress this a little bit here. Okay, many plants have those structures, and I think they look kind of nice. And of course, I'm talking about the so-called trichomes. The trichomes, and I have to, of course, change over to the microscope view. And let's have a look here, okay? And these here, let me focus this here. When I first saw those, I thought that these are some kind of spiders or insects, but these are not spiders or insects. These are so-called trichomes, and these are yeah, those star-shaped plant hair. Okay, a big bubble here, yeah. And what they do is in some plants they actually cover the bottom part of the leaf. Yeah. And if you find those, then yeah, you now know that these are not spiders, not insects, nothing dangerous. Even though they can look a little bit creepy, I have to tell you. But yeah, they're completely harmless. They are so-called plant trichomes, and I think these are one of the easiest and nicest to prepare samples when we talk about plant tissues here. Okay. Yeah, so, and as you enjoy more or less those plant here, let's have a look again at some of the comments here. Yeah, some people don't like flatworms though, okay? Yeah, and some people, yeah, have been sampling and found some interesting objects here, yeah. Question, can you please tell me, do you use some kind of magnifying glasses, for example, like dentists do, or something like that when composing specimens, especially that some are really hard to see? Yeah, I think so. This one is now not a question about the microscopes, but rather about magnifying glasses. Okay, you see that I'm kind of bending downwards, and I'm pulling out something from one of my drawers, and indeed, I can have a recommendation. And, yeah, magnifying glasses. So I'm going to switch over to the desk view over here. I can recommend. Yeah, those magnifying glasses. This one is a very old one. I still have it from the university days. It magnifies, it says here, totally of 10 times. As a matter of fact, I think it's even made of multiple lenses. And those magnifying glasses, of course, are used not only by biologists, but you can also look for them. They sell them online in Amazon and other shops for people who are into jewelry. So to test the quality of jewelry, yeah, these magnifying glasses are also used. Okay, and the way that you use them is you actually put them quite close to your eye, and then you kind of look at whatever object you want to look at. Okay, so yeah, and actually they're pretty good. Yeah. So if you are interested in magnifying glasses, I can recommend those. Yeah. And they actually are quite useful, and I occasionally use it as well. Yeah, if I want to simply check, for example, something on a specimen, if something is even worth observing. Okay, so yeah, so I go down the question. Yeah, some greetings from Costa Rica. Okay, some people don't like flatworms. Yeah, and some people like the live stream, which is always quite nice. Yeah. Could you later talk about LED replacement on the common introductory scopes? They're usually 1 watt, and I would like to add a 3 watt LED, mostly to get better performance at a do-it-yourself polarization. I can talk about maybe not. I can talk about LED replacement, maybe not in this very live stream here because I don't have anything prepared. But generally, it's not. It should not be difficult. LEDs are very low cost. And if you cannot get a 3 watt LED, then you just connect several 1 watt LEDs together. Just make sure that you have a small resistor at the beginning in front so that essentially the LEDs don't become damaged. Okay. Yeah. So some people want to see a little bit more about the Trickcom. So let's go back here. Okay, so can you zoom in into the center of the Trickcom? That would be nice. This one is now the 20 times objective, and I have to always switch a little bit the condenser setting here. And this is now 40 times. I always go up a little bit with the magnification as well. And then of course we want to focus a little bit here. Yeah. You can actually see that it almost looks a little bit hollow. You already know, some of you who have been in my channel for some time know already that I have DIC. So that means if you actually want to see it in regular bright field, then this is would be regular bright field, how most of you would probably see it. Okay, and then with the prism in here, that would be DIC, which I generally like to use for those live streams and for my YouTube videos, simply because that's color. And it simply makes the video more engaging. That's actually the only reason. Yeah, it's just a little bit more of a color thing, you know. If you want to look, let's have a look at the whole thing under phase contrast. I think I've never done this before. So this is now 40 times using, using DIC, using bright field. Okay, we've just seen that. And now let's do the following. Let's go over now. I have to switch all the way to my other objective. It's now 40 times. And I have to change to the phase annulus. And I have to open the condenser. And this would be phase contrast. Yeah. It doesn't look that much better. Yeah. Phase contrast, for those of you who don't know, is very good for observing very thin objects that are very transparent. And phase contrast converts a transparent object into one object where you have a black-white. Okay, so it converts the difference in refractive index into a difference of brightness. So not suitable for all specimens, especially thicker specimens are not very interesting, but I just wanted to show it to you here, okay? So, okay, I've heard you have 2 kids. How did you figure that out? I have 2 kids, yeah, about my age. I know I do look a little bit younger than I actually am. I just 2 weeks ago turned 51, honestly. I know I look a little bit younger than I actually am. Okay, how much water do you add to the PVA glue? It is more than estimated 10%. I tried it with about 25 to 30%, and it needs half an eternity to solidify under the cover glass. Yes, I think I added, I didn't measure it out, but I think I added around, it wasn't that little. It was more than 10%. I think I added around 20%. Yeah, I simply estimated this, and then I mixed it, and I let it stand for some time, and until it was properly mixed. And the drying time, when you say it takes a long time to solidify, then let me tell you that the Uperol, the mounting medium Uperol, which I also like to use, especially for mounting insects, it takes around 6 weeks to completely dry. So you have to keep the slide horizontal for 6 weeks in a place where there is sufficient air supply for it to dry out. Yeah. If you want to have really fast drying mounting media, then you can try those resin-based mounting media, which unfortunately also contain xylene, and they actually dry within a few days. But sometimes slow drying mounting media are sometimes better because it gives it enough time to actually go into the specimen. Okay. Do you have Instagram too? Well, actually I do, but I'm actually not using it very much. I used to put pictures on Instagram, microscopy pictures, but actually I do have an Instagram account, but have not been actively updating it for quite some time now. Yeah. So you'd have to see, I also call it Microbehunter, the Instagram account. Yeah. So the question is this a seed? No, these are not seeds. These are so-called trichomes. And these are plant here, and what I heard is that they kind of protect the plant a little bit from insects eating them. And I can imagine that because that's also sometimes quite common that, and we're going to look at this later on also in one of my other leaves, that it prevents, or at least not prevents, but at least reduces evaporation of water from the bottom side of the leaf. You see some leaves have a problem that they're losing too much water, and then the plant dries out. And if you have now the bottom side of a leaf covered with this fuzzy material, then the water cannot evaporate from the leaf as quickly. So there are sometimes, some plants have these adaptations to reduce the so-called the loss of water. Okay, looks like a glass flower, yeah, but it's actually the plant here, right? It's a trichome, yes. Okay, yeah. So I'm just skipping over here. Please don't show such scary things. Yeah. So this is the thing here. Yeah. So those plant trichomes, I just wanted to show them to you because they look kind of, I think they could look kind of nice. So that is part 1 of the live stream. Part 2 now is a little theory session before I'm actually going to start cutting up some leaves. Okay. I just want to do a little theory session now. And I'm going to use now, how do you say, some ready-made slides. and I'm going to show you now the following, the difference between a monocot and a dicot. And for that purpose, I'm going to go back and I'm just going to use bright field microscopy now. Okay, this is, let's go here. Okay. So this here is the cross section. I got a focus here. Okay, the cross section. It's one of my favorite ones, so you probably have already seen this before. The cross section of the of a sunflower. Okay, and I'm going to look at the so-called the vascular tissue, the structure here a little bit, and then on over here, this is the cross section of maize, corn. Okay, so this one here is a so-called a dicot plant, and this one here is a so-called a monocot plant, and you're able to see the difference also by looking under the microscope. And I need to explain what the differences are and what is even a dicot and a monocot. Okay, cells. So what we do here is this here is the cross section of the stem of a sunflower, which is a dicot. And I now need my computer mouse, and I now need to switch on the arrow here. So I have an arrow. Okay, I'm now able to use an arrow to point around. So that's of course the outside of the stem of the sunflower, and all of those little circles that you see here are of course individual cells. And again, this is a commercially prepared and I think a pretty high quality slide. I'm not able to get that type of quality together. I have to tell you, I don't have a very good microtome either, but yeah. And when you plant a sunflower into soil, what you have is you have two first leaves. Unfortunately, I don't have a demonstration here right now, but when this sunflower seed starts to germinate, there are two tiny leaves that come out. And that means it's a dicot plant. And those dicot plants have cross sections of stems where you have those nice looking patterns here. And what these cells are is these cells are the so-called the vascular bundle, and this is where water is transported up the stem. Yeah, so what we're looking here is at the cross section, and those cells here, those large cells here, that is the so-called xylem, because and the xylem tissues, these are the cells that transport water up the plant. And in a dicot plant like the sunflower, you see that those vascular bundles here that are responsible for transporting water up the plant are arranged in a circular manner, on the outside here. Yeah. And in the central part, you don't find, in the center here in the middle of the stem, you don't really find any vascular tissues that transport water up the plant. So what you have in here is you have actually the living part here on the outside. So there are also dividing cells that actually add layer after layer as the plant grows in thickness. And there's also the so-called the phloem out here, which transports sugar from the leaves down to the roots. Yeah, so that's a very typical arrangement of a dicot plant. And why am I telling this to you? Because maybe some of you, or maybe some biology students, are also going to listen to this live stream. And as a matter of fact, that is one of the syllabus contents that I also have to teach in school. Yeah, the differences of plant cross sections here. Yeah? And if you now look at a so-called a monocot plant, like for example maize or grasses, then you're going to, they only have one first leaf, and usually the leaves are very long and thin. Yeah, and if you look at the cross section there, then you're going to see that there are also those vascular bundles here, but they're scattered throughout the stem. They're not just arranged on the outside. Of course, there's some here, but actually those vascular bundles are all throughout the stem, and that is a typical configuration for a monocot. Yeah, but again, similarities are that you have again the xylem and the so-called the phloem, which are responsible for transporting water up. That's the xylem, and sugar down the plant. That's the phloem. Yeah. So that's so those large cells here that you see here are cells that transport water up to the up the plant. Okay, so I'm going to interrupt myself again, I'm going to interrupt myself again here and to ask a few more questions. Okay, question: what is the average lifespan of a cover glass? I have noticed that after a few uses and being cleaned, they get scratches. When I focus on the specimen behind, they're usually invisible. The average lifespan of a cover glass depends on how careful you are. Yes, I do reuse cover glasses as well. I do try to clean them. However, during the cleaning process, many of them break, and then of course I throw them away. So when cover glasses get scratches? Well, it could be, sure, but usually I would say that in most cases the scratches, because they are on the surface of the cover glass, they're not visible, right? Because actually you're looking at the specimen. Yeah. So if you want to look at the at what is on the top of the cover glass or on the surface of a slide, then yeah, you have to really defocus the microscope quite a bit. So usually those scratches are not so noticeable, and I think it's also not a big issue, because if there are things that kind of disturb the image quality, when you look at under the microscope, it's usually not the cover glass, just tiny scratches, but usually that there are some impurities in the specimen itself, or problems of specimen preparation. I usually think air bubbles, that those artifacts actually disturb more than at least small scratches on the cover glass. Yeah? Cover glasses can get scratches, not so much by regular use, but especially if you, what I like to do is if you, for example, throw your used microscope slides together in a container for collecting them, and if the glass rubs against each other, then this can of course introduce some scratches. Yeah, because some glass particles might also chip off. Yeah, yeah, generally it shouldn't be very disturbing. Yeah. What came first, a plant or a seed? So what's basically first, the chicken or the egg? And I will give you a biological answer here. Essentially, yeah, it could be, it was both evolved at the same time. Yeah? So it's basically it was an organism that was not a plant, and essentially evolved into a plant, and then basically slowly started to produce a seed. Yeah, so it's really the question, really, where does the plant start, or where does the seed start, and from what point do you actually want to count it as a seed? Yeah, there is the classical example about the chicken and egg question. What was first, a chicken or an egg? And the answer is very clearly it was the chicken, which came out of an egg which was not laid by a chicken. It kind of sounds weird, but there is, from one certain point onward, you have to actually define a living organism as a certain species, chicken. And then it was a bird. Before it was a bird, which was not a chicken. I know you think now we're kind of pushing out the problem a little bit, because where did that bird come from? Okay, yeah. So, yeah, nice coverage of trichomes. I study plants in scopes as a lot, and I see these all the time. Yeah. What do you use to clean them? The microscope slides and cover glasses. There are different ways how you can clean them. After I've used them, what I found out to be the most effective way is actually to use dishwashing powder. And then I found out it's not even necessary to actually use tissue paper, or you don't have to rub it off, because the dishwashing powder will handle most of it on its own. So I collect the cover glasses and the microscope slides in a plastic container. I add some hot water, and I throw in a little bit of dishwasher. So it's the dishwasher which I'm using for dishwashing machine. So it's not the liquid soap type of thing that you use for manually washing your dishes, but actually the powder they use for dishwashing machine. It contains enzymes, and I found this to work quite well. And then you of course have to rinse it. Okay. Yeah. Do only leaves have a stomata? Interesting. I think as a matter of, that's an interesting one. I think a cactus, for example, the body of a cactus, I think also must have stomata. Yeah, so, and it's not a leaf. Correct me if I'm wrong. Yeah. Question: how do I make a cross section of any plant leaf, turn it into a specimen? That's what I want to do actually today. Okay. Yeah. So I'm gonna do this right afterwards. Today I found a stomata on the stem of a flower, so I don't think so. Okay. I got sick today. Okay, good. No, not good that you're sick, but at least that my the live stream tries to cover a little bit, do away some of the boredom that you might have. Is cannabis the same type of plant? Of what? Well, I don't fully understand that now. Same type of plant as what? Okay. What are the very small cells near the edges of the dicot stem? It could, that's a good question. It could be, just a second, where are they? It could be also the cambium. Okay, so what you have here, I think it's like this, that over here you have the xylem, this here seems to be the phloem, and over here, usually in between it, you have the so-called the cambium, which is a layer of dividing cells. Okay, so if I don't, yeah, I've got this correct here, yeah. Brings back of memories of general biology. Okay, yeah. Do these prisms you use sometimes show true specimens color, or do they change it for better visibility? What type of objectives mode should be used to observe original color specimens? So the prisms that I'm using here right now, I'm using just regular bright field. So the colors that you see right now are the original colors. At least the specimen was stained, so it's not the totally natural color here. And the reason why you sometimes why I'm sometimes using DIC is not only for color, but sometimes you're able to see low contrast objects better. But I think for this type of specimen, it is not very suitable. Okay, so of course here now these are false colors here. Okay. Okay, so there's a suggestion to answer the questions which are unrelated at the end. Okay, that's certainly also a possibility. So, yes, because some of you have been waiting now concerning the preparation of some plants, and I would like to show a few things here to you now. First of all, I'm going to do the following. I'm going to just show you some simple hand cuts that I'm going to make. You need, of course, a cover glass and a microscope slide, as always. And I see that basically now all of a sudden the video interrupted here. Just a second, I need to try the following again. So here I am and the desk. Let me try this here again, because somehow the video got interrupted. And sometimes I have to restart the camera. That's interesting. Just a second. This never happened before. The mic. Look, I've completely lost my camera here. Just a second, guys. Look, I've completely lost my desktop camera. This is not, that's not so nice. Even though I've activated it, it's this is really a sad thing. You know what? I'm going to try to reinstall the camera now. Let's just a second. Okay, so maybe this helps. Ah, here we go. Okay, we're back. Okay, good. Yeah, so we're back here again. Okay, so for whatever reason my camera stopped. I had to unplug it and plug it in. Okay, no, we're back here. So let's try it. Let's try it again. Where is it? So what I would like to do now is over here I have a leaf. Okay, this here, yeah, of a maple. And what you need now is the following. Just to show you something that I was not very successful with, there are those cutting board knives, which you know. You can try those, but they are not quite as sharp as the razor blades. And so for this reason, I feel that, razor blades are better, but they are quite dangerous, obviously, for obvious reasons. So what I've therefore done is I've therefore made this plastic, how do you call this, plastic handle here. If you buy razor blades, then they basically come in little containers like this here. And these ones here are packaged in some paper for protection. And I'm just going to unpack one of them. And you got to be careful because, of course, they have a very sharp edge on both sides. Okay, so it is possible also to make cuts this way, but you have to be really careful that when you hold it, obviously, that you're not cutting yourself with the other side here. Okay, but it is possible, and I have already made cuts like this. Okay, and what you do is the following, and I'm not going to do that now with this one over here, just going to do it with this one over here. It is you got to, I always find it useful to kind of wrap the leaf around your finger. Okay, and then you try to make a very flat cut across here, and you want to go in and you want to go out again. Okay, so basically it's going to be thickest in the center and thinner on the edges, and we're going to be looking at the cells towards the edges because these cells are actually quite thin. Okay, yeah, so you have to really, I have a little bit of a problem seeing this. Okay, you see that I have over here already tried before to make a little cut, and yeah, and it's not, a little bit here. Okay, yeah, I got now a small part of the leaf here now on the top of the razor blade. I will now carefully try to transfer it on the slide, and what I generally, and it doesn't want to come down because of static electricity. Okay, here it is. And I found out that actually it's not cutting very well because this one is already a little bit corroded. Okay. So, and usually the more often you try, of course, it's a question of probability. Okay, here's another one. This one actually also worked. And I'm using now the bottom side of the leaf because I actually want to look at the stomates, the openings of the leaf. This is way too thick. Okay, I'm still going to try it because actually I'm not so much interested in the cells in the center, more on the cells on the side. Every time when you have small pieces like this, of course, there is always the danger a little bit that it starts to dry out. Okay, and for this reason, I recommend that you, of course, always add a little bit of water. Usually, sometimes I use tweezers. Okay, you simply add some water. And then we add a cover glass. Yeah, and these cover glasses are a little bit dirty because I did not properly wipe them. And then I accidentally had caught 2 cover glasses. So let's have a look now. Cover glass goes on top, and let's hope that we're able to see some of the plant cells and even some chloroplasts, hopefully this way. I mean, I'm doing this now very quickly, but generally, if you want to get proper pictures and so on, you really need to take your time with this, and then hope that you're able to find a few of them where it actually works well. Yeah, so let's focus. Okay. So, and we go now to the side, and we can already see the stomates. Yeah, here on the side. Yeah, this down here is a huge air bubble. The arrow is a little bit disturbing. Let's move it out of the way, and let's go up a little bit with the magnification here. Okay, and yeah, we can already see quite a lot. It might, I'm going to start explaining this. Let's find a place where where the cells where everything's a little bit thinner. Okay, so 40 times now. Okay. Yes, okay, and we are already pretty good to go, and let's have now a look at this. And the first check that I'm going to do is I'm going to use the fine focus, and let's see how many cell layers I'm actually looking at here. Now this cell layer is in focus, and I don't see another one. Okay, now it goes into focus. You can now see the cell walls here, are in focus. Yeah, the cell wall of the cells. I continue to turn. It goes blurry, of course, but I do not see another layer going into focus. So this actually means that I have one cell layer here on the side. Okay, and of course we can now see the individual cells here. And very interesting, of course, those oval structures here that you see here. These are the stomates or stomata. And these are openings in the bottom of the leaf. And what I'm going to do now is also here, over here on the top as well. And sometimes you're actually able to see here that there seems to be, a little horizontal opening. There are 2 cells called guard cells. So I'm going to go up with the magnification yet more. Okay. So, yes, let's move it a little bit into the center here. Okay, and then let's have a look at one of those stomates over here. In the middle here is an opening. Okay, and this is where gases, air, with oxygen can move out, because the plant produces oxygen by photosynthesis, and air with carbon dioxide from the atmosphere moves in by diffusion. And those cells here on the side are so-called guard cells. And what they do is when they basically absorb water, they will absorb water, and because they have an irregularly thick cell wall, when they absorb water, then the stomates will open up. Okay, so it's they absorb water, and this kind of deforms the cells in such a way that the stomates will open up and allow air to go in. And during night, when there is no photosynthesis, the stomates will close to prevent excessive water loss. Yeah, so this is actually a thing that is many plants have. Yeah, and that's a very now bumped into the stage. Yeah, and also very nice to see. But that's one of the things why I like plant cells so much. Is plants so much to observe under the microscope. It is because the individual cells can be seen so nicely. If you look at animal tissue under the microscope, because it doesn't have a cell wall, they don't have a cell wall. Therefore, it's difficult to see where does one cell start and the other one stop. I think this might be maybe 2 cell layers. No, still 1. And if you actually go now to a place where it's actually thicker, yeah, I don't know what that is, if it's some kind of a dust fiber or something else, yeah, then of course there's so many of them overlapping that you're not able to see anything meaningful. Yeah? And that is the reason why when you make a cut, always make sure that you're kind of cutting in at an angle so that you're able on the side, able to see those cells that are very thin. Okay. Yeah. So, yeah, just a second. I'm quickly again going through the comments. Have been okay. Okay. Your live feed looks clear if I view it signed off on another gadget. So maybe this one, yeah. So I'm just reading quickly through the comments here. Why don't you use scalpels instead of razor blades? Of course, I can also use scalpels to do that, but I find razor blades, they're pretty cheap, obviously. And I guess scalpels will also work, but razor blades do also have a little bit the advantage. They're a little bit flexible as well. So when you hold them, yeah. So in any case, this was number 1. You know what? Let's try the same thing here also on the other leaf. On this one over here. And now we can actually also let's try this one here. And if I happen to cut myself, I'm going to change the topic of the live stream from plants under the microscope to blood under the microscope. Okay. Again, you don't need a lot of force. And if it doesn't, and as a matter of fact, dull razor blades are actually dangerous, because they rip and then there's lots of friction and so on. Okay, again here. This is way too thick. Okay. So, but again, yeah, I cut all the way through here. Look at this. Yeah. So because the leaf is much thinner, I cut all the way through. So I'm still going to try it. So because I'm not so much concerned about the central part, but more on the edges. So maybe the edges are indeed quite okay. So let's try it again. Yeah, I mean, it does cut very lightly, but okay, here I also cut all the way through. Yeah. So it doesn't matter. We're going to give it a try in any case. I'm hoping that we're able to, of course. Up. Here it is. So. Take it away from my thumb and put it in here. And let's give it another try just to see. Maybe we're also able to see some of those stomates here. And some time ago, what I've done is, and I didn't want to do it today again, but it is also possible to actually put glue on the surface of the leaf and then peel off the glue and put the glue under the microscope. This way you're actually able to see the impression that the stomates made on the glue. So yeah, so let's go down here again. So let's just add this slide here, and now we have to have a look. This is much thicker, obviously. I don't know if we're able to find anything meaningful here. This is way too thick. Here on the side, maybe a little bit. Okay, here you see that's too much. There are not a lot of cells that are very thin here. Let me try over here. That's the other one. Okay. Let me go up a little bit with the magnification. Yep, that is a nice example of actually when you're cutting it too thick. Yeah, then unfortunately we're out of luck here. Okay. Yeah, because all of the cells cover up what we want to see. One thing that we might want to try is actually to look at the surface itself. Let's go up all the way with the brightness. Now it's too thick, unfortunately, and but what I have tried already, I made already leaf impressions using glue, and then you're able to see those. But unfortunately, as you see, yeah, this is not a good specimen. Yeah. And yeah, but that is the way it is. Too few cells here on the side that are thin. Okay, but one thing that you can see here, and that's maybe also quite nice, you see over here that the cells here are green, so they obviously contain chloroplasts and do photosynthesis. And then you see over here this long thing here is white without chloroplasts, because that is actually one of the vessels, the veins, the vascular bundle of the leaf where water is transported. And of course, those cells are empty, and therefore, yeah, they do not do photosynthesis, and therefore they do not contain chloroplasts, and therefore they're not green. Okay, well, yeah, that is a nice example. Here you can actually see some of the cells that are arranged, but it's too thick here, so we're not able to see the individual cells very well. Here now a little bit, yeah. But that is a nice example of not a very good specimen, because I'm not able to see those structures that I wanted to see. Okay. Do you still have the straight razor with a microtome to get that one sharpened? I know which one are you talking about? You're talking about, of course, this one over here. This is the most bad. I never got it sharpened because it was so bad. Just want to show it to you. Yeah, this one over here. Look at this. It's totally blunt and irregular and totally useless. Yeah. But that is actually the one that came along with the microtome. Yeah. So, you know what? But I mean, I hope you get the idea, and that some leaves evidently are easier to put under the microscope and cutting it than others. Okay. So let me quickly have. Yeah. Yep. Very quickly. Maybe. Okay. Look without water, only dry. How would the specimen look like without water, only dry? Generally, if you very often, if you do not add water, it will look too dark. I'm going to show this to you actually. Yeah, so that's the low magnification again. Yeah, and look, this one is the other one that I cut. You can see that it's much thinner. Generally, if you do not use any water, So let me try this again. Then very often the specimen look way too dark, and the reason is because there's too big of a difference in refractive index between the specimen and the surrounding air. Okay, so this is a little bit of a problem. So let's now try it like this again with the and try to make a very thin cut. Okay, I think this is way easier to cut than the other leaf. Okay, it's still a little bit too big, but I did not cut all the way through. So let's put it directly on a microscope slide again without water. I'm still going to put a cover glass on top simply to make sure that everything stays here. And what we expect to see, this is too thick, there we are. And what we expect to see now is actually a specimen which should look quite a bit darker than the other one. Usually this is what we normally have. Let me switch again to the microscope view. And well, actually, it looks also okay. Normally, it looks quite a bit darker, so that you're not able to see properly the structures. But what we can try to do, just for the fun of it, so this is now the one without water, and I'm going to just add a little bit of water now, just to try out how it looks like. Okay, get myself a pipette. So, I lost the tip of the pipette. Oh, that's interesting. I'll choose the other one. Just a second. Here it is. So, okay, I'm gonna add a little bit of water now. Let's see if there is a difference. Okay, so that's now with the water added. Yeah, I think the contrast is a bit better. It's kind of interesting to see a little bit how the water is actually taken up by the cells. Yeah, so a slight difference here. Okay, so I do also want to show you a little bit the microtome and how that is done. Okay, and let's again have a look here. I use single-sided razor blades. Yes, there are single-sided razor blades that have a actually also some that have only one sharp edge, and then they have a little bit of a reinforcement on the other side. They're a little bit thicker here, and therefore you can hold it better. Yeah. So as a matter of fact, those are probably also better to use, but simply they were more easily available because they simply sold them in the shop. For people who want to have a shave. Yeah. Okay. Okay. Do the cells of the stinging of the leaf look different? Okay. So yeah, what I'll do now is let's have a look now here at this microtome, and then I also want to show you some another way now to cut this here. And of course, I mean, you've maybe already seen quite a lot of times how I've prepared the potato starch grains, but actually today I'm going to use the potato as a way to hold a specimen in here. But just for the sake of completion, because some of you might not have seen this yet, I'm going to very quickly just demonstrate to you how potato starch grains can be observed. Again, I'm just doing this very quickly because some of you might not know that. Yeah. So you add a little bit of water here. You take a potato, and what you do is you simply scratch a little bit from the surface of the potato, and you dip what you've scratched into the water drop, and cover glass goes on top. Okay, here again I have 2 cover glasses. So cover glass goes on top, and then we're able to look at the starch grains. So that is a very simple way of actually preparing something. Just for the sake of completion, I would say. Yeah, and those round structures are the starch grains of the potato, right? And they look very nice in polarized light. And because I scratched the surface of the potato, I'm not able to see complete cells, but only those starch grains. They make a nice picture. Okay, so I just wanted to show it to you again, because it's usually the thing that you always want to show when you are dealing with potatoes. Now, those starch grains essentially are not a problem, obviously, right now, but I'm going to use the potato now as a holder for other plant tissues from a microtome. And this basically means that maybe there will be some of these starch grains also visible in the sample. If you don't want to use potatoes, you can, of course, also use carrots, which also works. Okay, so what I would like to do is I would like to make a cross section of this little stem here, okay, using a microtome. And yeah, so that's basically what I would like to do. And in order to do that, I'm going to cut a piece of potato like this. So it's almost like a French fry. Okay, and what I do is I make a cut in. It's a little too long, and I make a cut right in the middle here. So again, there are starch grains now all over the. Okay, there are starch grains now all over the place, and for this reason, you might want to use a carrot. But yeah, I didn't have carrots, so I'm going to use this, and I need a scissor. And we're now going to do the following. I'm going to cut this here. I'm going to cut this here. Okay. And this piece now I'm going to sandwich into the potato. So, and yeah, so it looks like this. And I'm going to now put it into the microtome, hoping that it's able to fit. And yeah, it has to go in here somehow. And it's a little bit too thick, and for this reason I'm going to now remove again some of the potato here from the side, otherwise I'm not able to get it in. Maybe also here a little bit from the other side. So, down here as well. And let's put this in again. And let's now try to put it in here. And I need to tighten the clamp. And I don't know. Okay. It's not held together properly here, but maybe it's going to work well enough for me to produce a decent result. And I'm not going to use those cutting board knives that I've used up to this point. I'm just going to use now again the sharp razor, because with that I had a slightly more success. And when you cut across it, then you have to make sure that both sides of the razor, of course, are on the platform. So don't cut it like this, because otherwise you're not going to get a very even cut. But something like this. Okay, so I've now removed the top part here. It's sliding out. That's not good. Okay, I need to turn I'm going to squeeze it a little bit more. I'm going to turn this now up a little bit, and let's see if. Okay, I've completely removed it, unfortunately. Okay, so here it is. The problem is that this was a little bit too, too unstable. So what I'm going to do is I'm going to simply put this whole thing here into the potato. Let's try it like this. Okay, another try. Sometimes, even if things don't work, you learn a lot. Okay, so yeah, you don't see this because I'm off. Yeah, so this is how it looks like, and let's give it another try. Okay, let's now put it in here again. Let's clamp it in. It should be a little bit out. Okay, and let's give it another try here. Okay, so now it's basically flush with the surface. And now I'm going to turn the microtome a little bit to turn it out. And let's try it now again. Okay, yes, now I got something here. So here it is. Yeah, maybe not quite as thin as I hoped it to be, but better than nothing. And we're going to now have a look at this here. Okay, put it on the microscope slide. Okay, does it? And I'm going to try it again. I'm going to turn it a little bit. And okay, here it is again. This time it's only the central part. Yeah, yeah, and just for the sake of demonstration, I'm going to now leave it as it is, add a drop of water, and again a cover glass. Okay, so let's have a look now at this, and honestly I have not looked at this before. So let's see how successful this is. Remove the slides. And let's put this in here. Oh yeah, it's a little bit not complete. And where's the other one? Here, it's a little bit better. Let's zoom in a little bit more and a little bit brighter. Yep, it's kind of, oh yeah, it's kind of okay. Let me see where the leaf starts here. Yes. Yeah, so that's basically where I have to focus again here. Yeah, it's still a little bit thick. Okay, so maybe let's have a closer look here on the other one here. This is a little bit thinner. And yet again, we can check the quality of the specimen. And because the individual cells are reasonably well visible, those bright spots that you see here seem to be starch grains from the potato. Yeah. So that's actually not a good thing, but yeah, who cares about that? We just have to, next time we just have to wash the specimen before putting it here, or we use a carrot. Yeah. And then again, we're able to see the different layers here, the epidermis, and so on. Yeah. And the idea is to somehow get the cross section thin enough that you're able to see the individual cells. And that is the thing that takes patience. So I'm reading the comments again. Is freezing specimens a technique to get thinner and more accurate cuts? Yes, there are. It's possible to do this as well. There are some techniques where you actually freeze the specimen, and then as the specimen starts to warm up, it expands. And it is simply the expansion of the warming specimen which is enough to actually allow you to cut with a microtome knife without actually having to advance the specimen, because the specimen will actually expand on itself. Yeah? If you're not planning on keeping the slide, do you always need water, or can you just put a cover glass on the slide? Well, generally with water the image quality is better. So this is really something that I recommend, and especially also these plant tissues. What they will do is they will dry up, and then they might actually shrink and deform. So actually it depends quite a bit on the specimen, but specimens that are naturally wet should be also kept wet. Yeah? So why not a leaf skeleton live stream? Honestly, I've made videos on leaf skeletons already before, and I was actually thinking about this as well. However, you have to boil the leaf quite a long time, and I didn't have time to do that today. Okay. Yeah. So, but I did make videos a few times, some months ago. Yeah. Yeah. I skim over the questions here. Could you cut the stem with a cover glass? No, this. I mean, I wouldn't honestly cutting the stem with a cover glass. I don't think that this is possible. Okay, because the cover glass is gonna fracture, and the cover glass is not sharp enough, and the cover glass is 0.17 millimeters thick. It's way thicker than a razor blade. Yeah, so it is using a cover glass is probably not the best method here. Yeah, the carrot is actually a possibility. Carrot microtome. What is the difference between a normal pipette and that special pipette that you use? Okay, I mean, let's put it this way. It's a question of convenience, rather. So what I have is a pipette like this, where you can adjust the volume. That is not the important thing, but it's kind of very convenient, and I like yeah, pushing this button, and it gives you a lot of control. Yeah, so it's a question of in my case, mostly a question of convenience. Yeah, but it's not necessary. Yeah. Does the potato skin cells differ from the inside? Okay, we can try that out. Okay, so so let's do the following. If you want to observe, so this was kind of the microtoming part. I also want to show you a couple of other techniques here. So for example, I've also done this before once, but not so often, so you might not know about this. But for some vegetables, this works quite well. Like for example, this little tomato. And what you do is you cut the tomato, and you take a fairly blunt knife. I know it's a little bit messy now here, but what you do is you try to kind of remove all of this pulp, okay, by scratching it, okay, until you get a very thin single-celled layer. I have to remove this pulp a little bit here. Okay. And again here, what I'm doing right now, this is not very, yeah, done very well, but I've really carefully done this for quite some time, and I got very nice results, which I'm now probably not able to reproduce in this live stream because I'm kind of doing this very quickly. I just want to demonstrate this a little bit as a proof of concept. But the thing about microscopy is that the specimen that you want to look at has to be sufficiently thin, sufficiently transparent, and sufficiently small. And if it's not like that, then you somehow have to get it into that form. Yeah, if I keep on doing this, it becomes a little bit more and more transparent. I've done this also with other vegetables that have a skin, and it also works. Okay. This might slowly work. Okay, it's now starting to tear. I'm doing this with a blunt edge, okay? So, yeah. So, and let's just give it a try now. Again, this is more just to demonstrate how this system works. I'm not saying that this is now the best result that I'm getting. Okay. Again, scissors. And now again, it has the tendency to roll up. So somehow we have to unroll it. It's not always the easiest either. Okay, but here we go. Might be rolled up a little bit on one side. And I don't know if this is not a single layer. It's rolled back on itself. So let me try it with my finger here. Let's see if this actually works now. So spreading it is sometimes the difficult part. Let's add water now. Okay, because sometimes the water actually does help to stabilize everything. So, and now let's have a look. And again, I think this is because I've done this so quickly now, maybe not the best specimen, but it's just a proof of concept a little bit. And we close the... As a matter of fact, it turned out better than I expected. Yeah, actually better. So yeah, you can see the individual tomato cells here as well. This allows you also a little bit to test your own quality of preparation. Yeah. So here we go. Here we see the individual cells much better. And 400 times. Okay. Yeah, so hope you get the idea here. The idea is always to kind of make sure that the specimen is thin enough, and then you can always do a focus check to see if you've got one cell layer or multiple cell layers. I think it does. It's actually one cell layer thick now. You know, this cell layer comes into focus and out of focus, and no other cell layer goes into focus. Okay. So I'll go through again some of the comments here. Okay, talking about the micropipette. Is that a 3D printed holder? Yes, it is. Yes, so this here is actually one that I made myself. I downloaded the STL file, and there are actually several of those. This is actually used if you have a something for cleaning, like a hot cooking plate or so, or some dishes where you've got some burned food on it, then you can use this to actually scratch off the surface. That's why it's actually for. Yeah. Okay. So cover glasses isn't sharp enough. Cover glasses aren't very delicate. Yeah, so we're talking here about cutting a specimen with a cover glass. Okay. Yeah, so, okay. Yeah, thank you for showing the stem part, although it's mixed with the potato cells. Yeah, unfortunately. So that's why I recommend that you try also other things. Now some people have been using styrofoam as well. I've also tried styrofoam. There is a huge disadvantage with styrofoam, and that is the styrofoam is incredibly tough on razor blades. That's kind of ironic a little bit, because styrofoam appears to be so soft, but styrofoam really makes the blade dull very quickly. And the reason is, it's quite interesting, it is when you cut with a razor blade through styrofoam, the styrofoam has so much friction that actually it will almost melt the very tip of the razor blade. As you cut through the styrofoam, heat will develop, a very small amount of heat, very locally, and this will actually melt a little bit the edge of the blade, and this causes it to become dull very quickly. So it's quite fascinating. But styrofoam, even though it appears to be so soft, it is very bad on those razor blades. So for this reason, I suggest that you use other things, and that's why I've been using potatoes or carrots. Yeah. Is there any schedule to live streams? Well, the schedule is the following. Pretty much almost every Saturday I'm doing a live stream. In one of the upcoming Saturdays I cannot do a live stream, beginning of July, because I've got an appointment, but I'll try to move it, but that's only on one day. Otherwise it's always at 9:30 in the evening Central European time. Yeah. How do you clean your pipette if it was used for contaminated substance? Okay. Glad you're asking. So the question is how do you clean a pipette like this? Well, simple answer, you don't. You just push this button here. When you push this button, the tip will be kicked off and you can throw it away. And then you have a box over here with fresh tips. Okay, that is the whole idea. But honestly, I don't do that very often because I normally use water for those tips, and water, of course, simply evaporates and I can recycle those. But actually, contamination is avoided completely by having those tips disposable. So just wanted to respond to that. Yeah. So very quickly again. Yeah. Then got it wrong. Okay. I recently looked at a paper yesterday which actually is used for root tip squash technique and actually perform genetic cytogenetic studies. Okay, yep. So, how to put a helicopter under the microscope. Wow, that's going to be a huge challenge. Okay, so we looked at this here, we looked at this here a little bit. There is something else that I wanted to show you. Of course, there is another preparation technique that I wanted to show you here, and that is also with the onion over here. I've done already many onion videos before, and of course, this is a little bit almost, I would say, overdone, the example. So I'm going to just show you how to prepare not the onion skin, which can be found between the layers, because you've already seen this so often probably with my videos, but I wanted to show you how you can actually take off the skin, the red part of the onion. Where is this? Here it is. I have to separate this. Okay, here we go. All right. How do you actually now take off the red part here? And that is also done by using a knife. And this, again, you do not want the knife to be too sharp, because you'll be cutting in here beneath into the flesh of the onion, and then you use your thumb to kind of press against it and to really stabilize it, and then you carefully rip off the top layer. Okay, and then you're able to see the red cells. Obviously, it's a little bit too thick again, because of the part here where I actually cut into the onion. So what I'm going to do now is of course, I'm going to cut off the thick part. Yeah, and you do it like this. Cover glass goes directly on top, and then I'll be adding a little bit of water as well. Sometimes it's easier to add the water afterwards. Yeah, and then you're able to look at the cells of the red onion. Yeah, again, that's the tomato preparation that I showed you. Let's remove this here again. And the red onion is over here. And again, we have to focus it a little bit. Yeah, and here are the individual red onion cells. Yeah, the round structures, of course, are air bubbles, which we just ignore, those round structures. Okay, so that's another short preparation technique. And so what I can recommend is that you, if you're interested in things like this, that you simply experiment around a little bit. Okay, let me quickly go through the comments again, and then I would like to show you again one of the commercial slides here, the cross section of a leaf, and then we're going to have a look at some of the leaf structures again. Okay. Can you do plasmolysis? Oh yeah, I could, but I don't have salt with me now. Basically, what you want to do now is you want to add some salt water, and if you add salt water, then you're able. It will actually remove. It will remove the water by osmosis, and the cell's contents will start to shrink. Yeah? That's actually something I didn't think of. But if you're interested in watching plasmolysis, I did make videos of this, okay? As a matter of fact, several videos using exactly those onion cells. So I would like to ask you to refer you to those because I don't have salt water with me right now, unfortunately. Yeah? Okay. Why are the cells pink? These are called, the pink color is called anthocyanins. These are simply water-soluble plant colors. And they're pink. Why? Well, because the cells have those pigments. Yeah, that's why they're pink. If you're asking about the advantage that the cells have, or that the onion has for being pink, unfortunately, I'm not able to tell you that. I don't know about that. Yeah. Here, that's interesting here as well. You're able to see that some of the cells are not pink, and the reason is as I ripped them off, the cells broke open, and the pink content of the cell started to flow out. So those cells here that are white, okay, actually are damaged, yeah? And therefore they've lost the pigment. Yeah? Staining request. Yeah. You ignore my comments. Well. Depends a little bit. What is plasmolysis? Okay, sorry about this. So basically what happens is if you add now a salt water or sugar water, I mean, I don't have it with me now, unfortunately, then what will happen is the cells will lose the water by osmosis. So the water inside the cell will go out, and you're actually going to see that the red part of the cell will start to shrink, as it loses the water. And this can be reversed again by adding clear water, yeah, and by adding water that does not contain salt. Yeah? And so this is called plasmolysis, is the loss of water by cells due to osmosis. Yeah? What I'm going to, there was another thing I just wanted to try out. I mean, I don't know, who cares? I'm going to try. Sometimes alcohol is also able to remove water. So what I could do is I could actually, just a second, where's the tissue paper? Here it is. I could actually try to remove the water here. Okay, I'm going to remove the water now. See that the air bubble starts to grow a little bit. And then I'm going to add some alcohol. And maybe this way I'm able to also plasmolyze the cells. I'm not so sure about this. Let me take this away, because I didn't plan this actually to actually show you plasmolysis, but in retrospect it wouldn't have been a bad idea anyway. So let's write a little bit. What I'm going to do now is but no guarantee that this actually will have an effect, okay? I've never tried this before, never added alcohol to red onions before, so I don't know what's gonna happen. Okay, so yeah, so here are again the red onions, and this is now completely dry. Okay, you see that there are actually 2 layers of cells now. I don't know. Let's give it a try. I'm just going to now add a little bit of alcohol here, and I'm going to know. So I'm going to now add a little bit of alcohol now under the microscope slide. I just want to see what happens. Yeah, of course the water and the thing mix. Look, the cells actually pop open because the alcohol dissolves the membrane. So I think some of the cells actually have popped open, and that's why it kind of started to spill some of the. Yep. You could see this maybe very briefly, that all of a sudden, yeah, the liquid started to, the red pigment started to spread. Yeah, no plasmolysis visible here. Oh, okay, was a nice try, but not really. Yeah. Why are some cells without color? Yeah, some cells are without color because as I've removed the onion skin from the onion, I've damaged some of the cells, and therefore the pigment started to run out. Yeah. So also, for example, if you look at, maybe we're able to see this usually on the side. Also, many cells on the side here are that have basically that are cut open. Yeah, also have lost, yeah, here, for example, have lost the pigment. You see this quite well. Yeah, cells that have been cut open, they've lost the pigment and therefore have lost the color. Yeah. And the other cells that basically were torn maybe on the top or bottom side, and therefore they've also lost the color. Yeah. The alcohol might decolorize as well. What could be, but what might happen is if you add a little bit of acid or base, because those plant colors often are pH sensitive, and sometimes they actually do change the color depending on pH. Yeah. Yeah. So I see the nucleus. Yes, that's correct. Yeah, each onion cell, those little dots that you see in the onion cells are actually the nuclei. Yeah. Really sure. Life plus smallest some. Yeah. Okay. Okay, let's have a look. Yep. Okay, so in summary, what time is it? It's also almost 1 hour, 1.5 hours again. In summary, what I just wanted to show you today. No, there's one thing, last thing I wanted to show you. It was the commercial slide of the leaf cross section. Again, a little bit of biology theory before I stop. Okay, because where is this? I have to find it. Lots of dirt and dust. Oh, just a second, see? Because I cannot look through it. I have no idea what that is. Just a second. Just a second. I'm quickly having a look under the microscope like this, because I need to find the leaf cross section. I totally lost it now. It's really strange. But it's here. Okay, that's a little bit strange. So bright that I'm not able to see it. Here we go. Yes. Let's zoom in a little bit more. I just wanted to show you a little bit how a commercially prepared slide looks like of a leaf. And how thin they are actually able to cut it with their microtomes. Because that is quite remarkable as well. Yes, I'm going to also try to briefly explain to you this here before I stop, because I've been already again doing the live stream for 1.5 hours. The question is now, what's the top and what's the bottom side of the leaf? Yeah, so this is the cross section. You can see the individual cells. The dots, the dark dots are the nuclei. And this bottom part here, that is actually the top side of the leaf. Now, how do I know that? Because do you see those cells here that are stacked vertically like this? That's called the palisade layer, and that is the top side of a leaf. Yeah, and the cells here on the bottom side of the leaf are much more irregular and much more loosely packed. So that's why we know that that's the bottom side. And this one up here, that is one of those stomates that we've seen also at the beginning. Yeah, we quickly, yeah. Yeah, so very typical here is those cells, and in the middle here you have, difficult to see a little bit, you have the opening. Yeah? So this is called the lower epidermis, and that's basically the thing that contains also the layer that also contains those guard cells in the stomach. And also over here, some people frequently ask, because they kind of wonder what this is. Maybe you're able to see this spiral-shaped structure over here. It's almost like those tiny lines that you see. It's almost spiral shaped. That's xylem. That is the cells that transport water up the plant. So maybe if we go up with the magnification a bit more, yep, here we see it much better. Yep, here we go. Okay, Those, if you see any structure that has those densely packed, spirally ring-shaped structure, it's most likely xylem. And these are strengthening rings that prevents the cells from collapsing and keeps it open so that water can be transported. Yeah. But again, I wanted to show this to you a little bit to show you that, yeah, commercially prepared slides, what they're able to do, what is very difficult to do if you actually cut them yourself. Because of the thickness of the specimen, and they use, yeah, of course, sometimes microtomes that are doing this completely automatic and in a very reproducible way, and then you are able to get very thin cross sections. Yeah? So very quickly, the remaining questions here before I have to stop. How do I find a mirror? How do I find a mirror on a microscope? Do you mean you want to connect a mirror to a microscope? Well, there are some companies that offer them. Okay, not all microscopes offer them, but yes, there are certain microscopes around that come with instead of a lamp with a mirror, because sometimes you want to use them in the field, for example, where you do not have electricity. Yeah? Is DNA visible under the microscope? The answer is yes and no. I have to be very clear about this here. DNA, the individual DNA molecules are way too thin to be seen under the microscope. However, if you extract the DNA and if you put it under the microscope, you are able to see those long strings, because these are probably hundreds, if not thousands, of DNA molecules bundled together. So you are able to see as the string-like nature, but these are not individual DNA molecules. Sometimes there are also proteins associated with it. It kind of makes it thicker than it actually is. And under the electron microscope, under good ones, you're even able to see the individual, barely you're able to see the individual DNA molecules even, because they are so thin. Yeah. So no, DNA is not visible under the optical microscope. Generally, that is correct. But I have made videos and I was able to see this string-like nature under the microscope, but these are not individual DNA molecules. Okay. As a matter of fact, even if you extract DNA and if you take a little wooden toothpick to pull out the DNA, you're able to see the strings, the string-like nature and texture of the DNA, but these are not individual molecules. Yeah. Yeah. I can't find the picture on the microscope. Can you help me? I don't understand what you mean. You mean you cannot focus it properly? That you're not able to see something? Yeah, that's something that it's not so easy to explain over a live stream. Yeah. But what I recommend is that when you start with observing, that you always start focusing and observing with the low power objective first, and then when everything is in focus, you work yourself upwards with the magnification. Yeah. So have you seen stomates opening and closing before? Not under the microscope. Okay, because this would actually mean that I have a single cell. Yeah, I'd actually have to be able to observe the surface of a leaf this way, but as soon as you make a cut and a section through a leaf, you're actually destroying the leaf, and then yeah, they might not be able to open and close before. Yeah. Where are the phloem cells? Depending on what you're looking at, sometimes associated next to the xylem. Yeah, so the phloem actually does look different. The xylem is actually has much larger cells. Yeah. You can also try looking at the vascular bundle and locate the phloem and xylem to determine the leaf orientation. Yes. Okay. I'm gonna do Okay, there are a couple of students here as well. Does an electron microscope use only one lens? If yes, what magnification lens? Well, electron microscopes don't use any optical lenses. They use magnetic fields, and they have actually several coils in them that generate an electric field. So you call them kind of like, I don't know, like magnetic lenses if you want to. And electron microscopes in that sense cannot really be compared with optical microscopes, but electron microscopes can magnify up to several 100,000 times. Optical microscopes only 1,000 times. Okay. What microscope do you use? Well, I have several microscopes. I use essentially optical bright field microscopes, and I would say that even relatively low cost microscopes can already produce quite acceptable results. Yeah? Can you get a sample of blood that has the COVID injection nanotechnology in it? No, I can't, because honestly, there is no nanotechnology in it. If there were, we'd be dead already, because any foreign matter in the blood vessel would actually clog the blood vessel and would cause circulation problems. And yeah, it means if there were any nanotechnology or any particles, solid particles in it, then it would, yeah, we would already know it even without a microscope, because we would have circulatory system problems. We don't want to have any solid particles floating around in your blood system anyway. Yeah, so there is actually no point investigating things where we already know that this is not the case. Yeah. Have you done a DNA excretion of a banana at home? Yes, I have, and I've also made a YouTube video. In this very channel, if you search for DNA extraction, you can actually find it. Yeah. Can you get some dental anesthesia to see if it's self-assembly nanotechnology in it? Why should there be any self-assembling nanotechnology in it? Yeah, and what even is that? Yeah, electron microscopes don't really have much to do with light. That's correct. Okay. What kind of microscope is that? Well, I have an Olympus microscope right now. My first Olympus microscope I got about 1998. It was an Olympus CH40. It was a bright field microscope. Yeah, and I've got one as well right now. I just bought a Multic microscope in March, and I like it a lot. Yes. Okay. Do you like, I don't know, the L&W scientific microscope with the Apple front? I don't know. There's so many brands out there. Yeah. And I would say if you're happy with the microscope, then yeah, it's doing its job. What are some microorganisms that fascinate you the most? That's an interesting one, really. I think all microorganisms have interesting aspects to it. But if I want to answer it like this, I mean, I'm a bacteriologist. I studied bacteriology at university. But I consider ciliates and flagellates, the eukaryotic, the protists, in other words, much more interesting to look at under the microscope, because they're larger, and you can actually look into the cell to see what's going on inside the cell. In that sense, I consider bacteria by far not as interesting under the microscope, because they're significantly smaller and they don't have quite as much information content. Yeah? So generally, I would say that under the microscope, if you're asking for microorganisms, then I would recommend that you have a look at protists, and you can find plenty of them in water samples. If you can repeat what brand and model should I write down to buy? Honestly, there are so many. Honestly, most microscopes are made these days in China, and then they are imported into Europe, to the United States, and so on, and they're rebranded. So if you have, for example, Swift and Multic and Amscope and whatever they're called, I would say most of them are manufactured in China. This is the place where most of the products are made, and then they're rebranded. Yeah, and this says nothing about the quality. It depends really much on the individual microscope that you want to buy. And one of the difficult things is that different people have different expectations of what is a good quality microscope, because what is good quality for one person is not good for the other person. But I can just reassure you that these days, pretty much, I would say, in the standard range, if you buy a microscope for 400, 500 euros or dollars and upwards, generally will produce good enough results for you to be satisfied. But if you're a complete beginner, I sometimes suggest that you don't spend a lot of money. Right at the beginning, you just get one, maybe a basic introductory microscope, and then as you learn, you can always upgrade. Yeah. Yeah, I mean, if you really want to have good microscopes, good under quotation marks, then go for Zeiss, Leica, Olympus, Nikon. But I guarantee you almost won't be able to afford them, because they're so expensive. Yeah? And it's also not necessary sometimes, because these companies produce microscopes for the research market. And so therefore, I would say that I would say that microscopes in the range of, let's say, 400 euros or dollars and above are quite fine. Introductory microscopes can already be obtained for around $100 or euros, and that's how I would start probably. Yeah. Yeah. So, and I measured microscope magnification with a microscope ruler, and I tell you that microscopes don't magnify anything 1000 or 2000. They magnify maximum 100 times when used with an eyepiece. Well, I will tell you the following. There are two different kinds of magnifications. There's so-called linear magnification, and also one's called linear magnification, the other one's called magnification. So the magnification that you get when you look through an eyepiece is different than the magnification that you get when you take a picture of something. Yeah, in both cases you use the word magnification, but actually the math and what it means is actually different. Yeah, so I can imagine that maybe this is relates here a little bit here. Yeah, so I think people, I'm going to leave it again at that. It's 1 hour and 40 minutes that I'm already talking. Yeah, and what I would like to do now is I'm simply going to, look at this, I have to start cleaning up my desk again a little bit. It's a beautiful mess that I have now, but that's okay. So I usually start cleaning up everything again. And yeah, I think if you have any recommendations on what I should do, then yeah, please write something in the comments. I've been actually thinking, because some people again asked of actually doing some DNA. Some people want to know how to do DNA extractions. And honestly, I was thinking about doing that. But then again, it's not really microscopy related. So it's really a little bit difficult. Well, then I say maybe I'm just going to do it anyway because the interest is here. So I'm thinking about this. Maybe I'm going to show you how to do a very simple DNA extraction, which can be done at home with basically household materials. And let's also try to put it under the microscope in any case, so that we're able to see those strings. Even though these are not individual DNA molecules, we're still able to see some basic structure here. Yeah, so maybe that's what I'm kind of thinking for next week, but I'll see. Okay. Yeah, I wish you all the best, folks. Hope that you enjoyed it again. Hope to see you again soon. And yeah, see you around next time. And bye-bye. All the best.